Hello and welcome to part 3 of 4 of the Science 30 presentation for meiosis, covering topics in Unit 2 or the biology unit. During prophase 1 of meiosis, homologous or similar chromosomes come together through a process known as synapsis. During this time, the chromosomes are so close together that this process called crossing over can occur. It occurs when these chromosomes are touching. At that point of crossing over is called a chiasma, from the ancient Greek chimera, or combination of two different things. This leads to genetic variation since the sections of DNA are exchanged and result in the mixing of maternal and paternal genes. The result is two chromosomes that are different from the original. So you have bits of yellow in the green and bits of green in the yellow. During anaphase 1, the chromosomes are separated in a process known as segregation, and each chromosome exchanges DNA. So here we have the difference between meiosis and mitosis. In mitosis, you have two chromosomes that are paired. They line up in a straight line on the metaphase plate, and one red one and one blue one are each pulled into the resulting cells. So the end result are two cells that are identical to the original. So two diploid cells that are identical to the original. In meiosis, you have DNA being replicated. You have these chromosomes pairing up as a tetrad. Crossing over occurs, so blue and red get mixed. One half of this tetrad is pulled into opposite sections because this is one chromosome that is being pulled this way and one chromosome that is being pulled this way. The resulting cells are 1N, not 2N, because you have two different chromosomes, 2N, only one on each side, 1N. Through the second meiotic division, you have these chromosomes being pulled apart. So you have one chromosome being pulled this way and one being pulled the other way, which results in 4, 1, 2, 3, 4, 1N cells, which do not have the same DNA as the parent cell. And even here you have some crossing over happening, mixing of blue and red DNA. Again, notice the difference between mitosis and meiosis. In mitosis, you have 4 DNA molecules. You have 4 DNA molecules, or 4 sister chromatids that you start out with. These condense and are lined up on the metaphase plate. Each one of these pairs of chromatids are pulled apart, resulting in 1, 2, 3, 4 identical chromosomes in each cell. In meiosis, you have the same thing occurring. In interphase, you have chromosomes condensing, but instead of floating free, they line up in this these tetrads, where homologous chromosomes are paired together. These pairs are lined up along the metaphase plate, resulting in 2 chromosomes on this side and 2 chromosomes on this side, instead of the 4 that you started with over here, 1, 2, 3, 4. So this is 1, 2, 1, 2. This is a 1N cells. Each of these pairs of sister chromatids again line up on the metaphase plate, and each sister chromatid is pulled to opposite sides of the cell, resulting in 4 cells with half of the DNA of the parent cell. So 2 chromosomes instead of 4 chromosomes. Again, here is another example of meiosis versus mitosis. In meiosis, synapsis and crossing over occur, so you have those tetrads coming together. In mitosis, there is no synapsis or crossing over. In meiosis, the homologous DNA molecules align independently, and in mitosis, the chromosomes align one by one along the metaphase plate. Homologous chromosomes separate and daughter chromosomes separate. So you have pairs going in opposite directions over here, and you have one going in opposite directions over here. The daughter cells have two pairs of chromatids on this side, 2 pairs of chromatids on this side, as opposed to the 4 pairs that were up here. So this is it goes from 4 to 2, so that's from 2N to 1N. These pairs of sister chromatids are separated, so resulting in 1, 2, 3, 4 daughter cells that are not genetically identical and have a 1N complement of DNA instead of a 2N complement of DNA. Mitosis produces daughter cells that are genetically identical. One of the key outcomes of meiosis is to produce daughter cells with different combinations of genes. This variation is accomplished by gamete success, crossing over, which depends on the distance between genes and the length of chromosomes, independent assortment, or the way one homologous pair lines up has nothing to do with the way the other homologous pair line up. In crossing over, homologous chromosomes pair up, and then this chiasma is formed. Recombinant, this chiasma is resolved, and the result is what they call recombinant chromatids, or chromatids that contain DNA from one parent and the other parent. Again, here's another picture of crossing over and recombination. You have this chiasma is formed. Here is your recombinant chromatid. Each one of these are pulled into a separate cell during meiosis 2. The likelihood of crossing over is greater if the genes are farther apart than if they are very close together. And that's one of the ways geneticists determine whether or how close genes are. If you see a lot of crossing over, you can surmise that the genes are farther apart than if they're closer together. Crossing over recombines or mixes. Recombines or recombination is just mixing. So crossing over mixes genes between sister chromatids. And here's a picture of chiasmas occurring. So these are all locations of recombination. Independent assortment just means that it has, there is no rhyme or reason or order of lining up these chromosome or sister chromatids. You're not going to get all blue on one side or all green on the other, or all father or all mother. You're going to get a random mix of mother and father, and it doesn't matter on which side these things are.